diagrams, these novel features and regressions, I want to focus a little bit more upon just the big scale hierarchy and kingdoms and domains. These are the broadest groupings that we have, and just discuss a little bit of the historical progression and where we're at today for the most part. There's still a lot of debate today. I don't think our current system will hold up indefinitely, and I'll kind of explain why that is. So starting off, we had plants, animals. Big stuff, we can see it. These were our two kingdoms. After this, as we got more and more evidence, as we got more and more data with things like microscopes, we eventually shifted to what's the five kingdom system. And so we had animals and plants still, but we added in fungus, which previously they used to lump into plants, but they realized they're not plants. We have protists, which are going to be eukaryotes, but these tend to be unicellular eukaryotes. These tend to be eukaryotes that are the ancestors of plants, fungus, animals. So they're essentially a kingdom of exclusion, where if you're not a fungus, animal, or plant, but you are a eukaryote, you belong in Protista. And this is the problem with Protista, this is the current controversy, is everybody says Protista is not monophyletic. There's different lineages. You've got the plant-like guys, you've got the fungus-like guys, and animal-like guys, and you've got some guys that are just a bit weird, that are different than stuff. And so pretty much everybody agrees that Protista shouldn't just be a kingdom by itself. That this whole grab bag, idea of a kingdom doesn't function well when we're looking at things cladistically. The problem is just deciding how to break it up. So some people want to split it into three things, some people want to split it into 5 different kingdoms. But for now, just understand that we're going to keep Protista together, but it's going to be kind of under protest where everybody agrees it needs to change. They just can't agree how it should change. And then we had Monera, which would be the prokaryotes. So these ones were obviously different, but because they were so small, we could tell that they lacked these membrane-bound organelles, but we couldn't really see a whole lot more beyond there. And so it made it really hard to distinguish amongst prokaryotes, so we just kind of said, oh yeah, they're really simple, and lumped them all together. And so 1 kingdom for prokaryotes, 4 kingdoms or more, as we fix it eventually, for eukaryotes. Now, as we got molecular evidence, this molecular systematics, DNA, proteins, looking at the various molecules that we use, we realized that prokaryotes are more different from one another, at least the major groups of them. There's the archaea and bacteria, sometimes called archaebacteria and eubacteria. So as we looked at these guys, we've realized that molecularly, they are way more different than one another than any eukaryotes are. So we started to see that these eukaryotes are all fairly similar when we look at their molecules, and we notice these prokaryotes, specifically archaea and eubacteria, are very different. And so this caused a problem, because when we look at the 5 kingdom system, or the next thing they did was make it the 6 kingdom system, they added 2 kingdoms, they split Monera up, we still look at this and see, well, there's 4 or more kingdoms of eukaryotes and 2 prokaryotes. So based upon that, just doing simple math, eukaryotes appear to be more diverse than prokaryotes. But prokaryotes, with their 4 billion years of evolution, are molecularly way more diverse as a whole. Each of them individually might be fairly simplistic, but as a whole, they're tremendously diverse. They can do things that eukaryotes cannot dream of. Fixing nitrogen. You've got methanogens, you've got extremophiles that can do things that eukaryotes just don't or can't. And so we had to have some way of kind of representing this diversity appropriately. And so that's where the idea of domains came from. They said we don't need to scrap the system. This whole kingdom, phylum, class, order, that's fine. What we'll do is we'll just add something above a kingdom, and we'll call it a domain. And so that's where we got the 3 domains. And the domains show that prokaryotes are more diverse because the domains have 3 of them, and 2 of those are prokaryotic. So archaea, the kingdom, belongs to archaea, or archaebacteria it's sometimes called, the domain. Eubacteria, or bacteria, the kingdom, fits into its own domain, bacteria or eubacteria you might see. And then all of these other guys, Protista, Plantae, Fungi, Animalia, if we break up Protista, all those ones we break it up into, all of those fit into just a single domain, eukarya. So this represents the fact that prokaryotes have a lot of diversity. We've got archaea and bacteria, whereas eukaryotes have much less diversity. So I'll just kind of write it small if you can even read it. But they've got much less diversity between them. So that's where we're kind of at today, is we've got these three domains represent the molecular diversity very well amongst prokaryotes and eukaryotes. So right now we're just kind of arguing, trying to get down to what is the best monophyletic way to represent the rest of the kingdoms. And so at least for right now, most of the debate revolves around how we should break up Protista. All right, so as far as the tree of life, I just want to go through and bring up a lot of the molecular evidence that we used for stuff way back when is to look at the ribosomes, specifically the rRNA that makes it up. Because this stuff is ultimately going to be highly conserved, which means it changes very slowly. And so this is very useful if we're looking at stuff that might have diverged billions of years ago. Whereas if we're talking about something that has diverged relatively recently, we would want to use something that's a poorly conserved gene, meaning it rapidly changes. So you might look at like eye color or something where it doesn't have as big of an impact on your survival. But for rRNA, it's pretty important stuff, so this tends to change very, very slowly. So when they talk about molecular evidence, just keep in mind this is a very good example to use. The rRNA genes, the genes that ultimately code for making the ribosomes that you have. And prokaryotic ribosomes will be different from eukaryotic ones. And archaean ribosomes are actually more similar to eukaryotes ribosomes than eubacteria. And that's one of the things that we see, is it does appear as though that we are more closely related by quite a bit to archaeans than we are to eubacteria. You know, there are some archaeans that have introns that have some proteins with their DNA. Their ribosomes are more similar to ours. So there are specific traits that archaebacteria or archaeans have that eubacteria or bacteria don't. And so that prokaryote that kind of eventually became the first eukaryote was most likely, very significantly more likely, to be an archaean. Now there is problems though, because as we're going to discuss in a little bit here, prokaryotes can be a little bit loose about their DNA. They can pass stuff around, even across species lines. And so because of that, we get this horizontal gene transfer, where an archaean could go through conjugation and trade DNA with a bacteria. Or they could pick up some bacteria that's from a dead organism, whatever it is, eukaryote, prokaryote, and use it. And so this DNA is not stuck in this vertical pathway where you can only get it if it's passed on by your parents to you. And so that can make things difficult too, because we can see stuff jump back and forth between some of these things. And so it makes it harder for us to track everything with what went on way, way back when. So evidence is pretty solid that this is the appropriate way to represent it. So you can see bacteria is off on one branch. From there we got archaea, and archaeans we see are more closely related to the eukaryotes. But there is different ways of doing this, and you'll see these are all just three different versions of how this might have went down, but you can see in all of them you still have this kind of separation between the eubacteria and the archaea that very early on this split occurred. Some of the earliest bacteria might have been archaeans, but this split occurred early on to give us eubacteria as well. And so this is just showing three of the possible ways that that might have happened. I'm not going to make you memorize all of them, but just be aware that we're still trying to piece together exactly what happened way back then, even though we have at least a pretty good idea that we had this early split between the two types of prokaryotes, and that the archaeans are the ones that ultimately gave rise, at least in large part, to eukaryotes. There are some eubacteria like cyanobacteria that became chloroplasts and other plastids, so it's not like they're not part of our history at all. But as far as the original cell that was a eukaryote that developed a nucleus, etc., that was likely going to be what used to be an archaebacteria. All right. So as we transition just a little bit to get into some of the prokaryotes in general, I want to make sure you guys realize these guys are incredibly small, especially versus eukaryotes. So an average size of a bacteria would be around 1 micrometer. They can be smaller, they can be a bit bigger. The average size of a eukaryote, we'll say, is around 50 micrometers, so much larger. They can be bigger, they can be smaller, but I want you to realize there's a huge difference here. And that's why the prokaryotes are so much more numerous. On your body, if we just kind of look in and on our bodies, we're outnumbered by prokaryotic cells by about 9 or 10 to 1. And the reason for that is because they're so tiny. I mean, if they're 1/50th of the size, it's easy for them to coat something. You can have one eukaryotic cell and just have tons of these bacteria that can live on it, or if it's endosymbiotically, I suppose, they could even live inside of it. And so that's useful to kind of think about that if you grab like a chunk of dirt, if it's very fertile, rich with organic matter, you can have a ridiculous amount of bacteria in that soil. Way more than all of the people in the human race, possibly more than all the people that have ever lived that are human, period. I mean, there's a ton of stuff that can be just in that small amount of soil. There's a ridiculous amount of bacteria, and a lot of that's because they're small. Now, other things, when we classify them, it's difficult to look at them because they're so small and use a lot of structures. So some of the simple things we'll do, which I don't know if I'll actually quiz you on, but it's useful when we talk about their names, which you might see come up periodically. They're going to have three shapes. So anything that's a cocci shape, that's going to be round. So these guys are going to be cocci. All right? And that's like streptococcus. You'll see a lot of their names tell you what it is. You've got some stuff that, like Bacillus anthracis, they have a bacilli shape or a rod shape. So Bacillus anthracis, the stuff that causes anthrax, looks like it's rod shaped. It looks kind of like a sausage link or hot dog. And then there's helical, which are oftentimes referred to as spirochetes, are the guys that do this. And that's like the guys that cause Lyme disease and syphilis. So these guys look like a pigtail. So those are the three shapes that we use to classify them. Now, as far as structures, most bacteria will have a cell wall, and that cell wall will contain a special structure, in most cases at least, if it's a bacteria. Archaeans, you'll see, do not use peptidoglycan. It's one of the reasons that we are likely related to them. But peptidoglycan is just a fancy mixture of sugar and protein that makes up much of the cell wall of bacteria, specifically eubacteria. And gram-positives have a lot more of this, so they happen to stain a specific way. So when you gram stain them, they turn to be purple. So these guys are gram-positive. The guys that stay red when you do a gram stain are going to be called gram-negative, because you initially stain them red, then you add this darker stain, and so if they don't stain, if they're negative for the stain, they're gram-negative. Most of the pathogens, the things that cause you harm, and the things that are harder to treat with antibiotics are gram-negative. So if you do have an infection, it's typically better if it's gram-positive because it's easier to treat. But that's one of the ways that will also differentiate them. It's not just about their shape, although you can see this is cocci. You can see these guys look like they're bacilli. But aside from just using shape, we can also use our ability to dye them, which is really just testing to see what their cell wall is made up of. Gram-negative guys tend to have a lot less peptidoglycan. They have more of these things called lipopolysaccharides. You don't have to memorize that, but it's just a different structure. Many of them also have this coating, this kind of, I guess you could say it's almost like a jelly-like coating called a capsule, which can help protect them. Just like our stomach lining has a layer of mucus, you can think it's somewhat like that they can line themselves. And so this can help them adhere to stuff. This can help protect them. Just like they also have these pili, which are these extensions that you can see here. And these pili can also be used for like adherence to stuff, and they can be used for transfer. You'll see that when we talk about conjugation, when they trade DNA, they can do so using these pili. So it'd be kind of like if you take your fingers and stick them together and merge them. That's what the pili do, and then you can let stuff transfer back and forth, which in their case is DNA, not like STDs or something. All right. Form and function, really crash course. I want you to remember that when you talk about prokaryotes, they will have organization. So the nucleoid region is the part where they keep their DNA. It's not membrane bound, but they still have a special place for it. Plasmids will be these small circular pieces of DNA that they can use to kind of move around their genetic information, both within the cell and to other cells. Endospores are kind of sweet because the cell can kind of become inactive and pretty much die temporarily, and then it can come back later. But there are cells that have been revived that are bacterial that appear to be tens of millions of years old that they've been able to bring out of this state as an endospore. So it's really, really cool, especially if you're big into like the whole seeding of life, where stuff could get caught on a piece of debris because something hit and ejected it in space. This is really useful for that. It's really also cool if you like diseases, because some diseases can form endospores, which means they can spend like indefinite time just chilling out places, waiting for you to get some on you and cause problems. Tetanus is a good example of this, so it can just kind of sit around in the soil, sit around on metal pieces, especially rusty metal. So that way, if you get a bad cut, or like in wars, they'd have wounds and they'd be on the ground, and then they'd get lockjaw tetanus. And that is obviously bad if you get it, but if you're just looking at disease and thinking about cool ways they manage to spread, then it's kind of cool. Asexual reproduction is what they are typically going to practice. They do not have a strictly sexual form of reproduction. They're going to undergo binary fission, which is like a simple version of mitosis. It's the ancestral version of mitosis, most likely. And so this will just be more or less copy your one circular chromosome, move it to either side of the cell attached to the cell membrane, and then pinch apart or build a wall in between, kind of like we've seen before, so you have two cells. So it's pretty straightforward, it's simpler, you don't need spindle fibers or any of that stuff. And they can get away with doing asexual reproduction a lot because they reproduce so quickly. And because they reproduce so much, so rapidly, they end up having enough mutation, because even if the rate of mutation is low, if you do something enough, it's going to happen. You know, if you buy enough lottery tickets, you should win unless you are really unlucky. And so humans who take forever to reproduce, 9 months just to get a kid, even after you've had it start, whereas bacteria, you can be talking 20 minutes and one becomes two. 20 more minutes, you've got two became four. So theirs is so much astronomically a higher rate of reproduction that they can rely on mutation to provide most of their diversity. But not all. So bacteria do have their own way of kind of having sexy time. It's not the exact same, but it's close enough. And the three ways that they can have this mixing up of genetic info, is transformation, which is where they kind of grab naked DNA, like pieces of DNA from dead cells, things that are no longer intact. They can grab that DNA, bring it into their cell, and use it. So we can't do this as eukaryotes, but they can kind of like look around and be like, oh, that's a cool gene sitting there, and take it. Conjugation is when they use these pili, and they can connect it between bacterial cells, and then they can pass through these plasmids. And so they can essentially trade plasmids. And these plasmids can carry stuff, like they call it the F plasmid, the fertility plasmid, that allows the cell to do this process, because this process isn't always like consensual. It's like one guy just kind of sticks somebody else and then transfers stuff. They can also be R plasmids you might see, which carry antibiotic resistance. This is why it's kind of a big pain, because once some bacteria develop antibiotic resistance, they can transfer that to other bacteria of their own type as well as other types. And so it makes it fairly easy for antibiotic resistance to spread amongst a variety of different types of bacteria. So that's conjugation. This is probably the closest thing to bacterial sex we have. And then transduction is going to be one that involves viruses, where when a bacteriophage, a virus for bacteria, infects it, sometimes they grab genes as they come out. And so they don't just grab their own DNA, they get a bit extra. And so when they infect another bacteria, they add those genes to that new bacteria's genome. And if they are a lysogenic bacteria, one that kind of hangs around for a while, if not indefinitely, those genes now have become part of the new bacteria. They've been added to its genome. And so this is another way, the third way, that you can get new mixes that don't just have to do with mutation. All right. The last thing we've got here is I want to briefly mention archaeans, because we've really been focusing on eubacteria, because those guys are the ones that most commonly cause disease. They most commonly live in the conditions that we're in. They're the ones that we most commonly interact with. But these archaeans, which tend to be extremophiles, which just means they like harsh conditions, halophiles, for instance, like lots of salt. So this would be like the Dead Sea. There are some of these guys that can live there, though eukaryotes and eubacteria would not. There's thermophiles that like high temperature, so they can live in hot water heaters. They can live in hot water springs. Many of these, like volcanic springs. And so these guys can survive temperatures, in many cases, up to and around boiling. I mean, these guys can tolerate a lot of heat in many cases. There's also methanogens that you can see that tend to be anaerobic, living in swamps that produce methane. So you get like the old swamp bubbles of methane that you'll see in movies and such, because it looks cool and spooky. And that's ultimately where that comes from, is there are some of these archaeans that are methanogens that tend to live in these anaerobic areas like the sludge of swamps. And one of the byproducts of their metabolism is that they can produce methane. There's some other guys too, I believe, that actually use methane to go through and do their metabolism, but those are some other chemosynthetic guys. But needless to say, the Archeans have a lot of really cool guys that live in these kind of weird circumstances, these harsh circumstances, which is why a lot of people think that Archeans might have been the first cells, because the early Earth resembles a lot of the conditions that we see Archeans still live in today. So these guys may have, come around and existed and been like, this world is amazing 3.5, 4 billion years ago, when the Earth was really not that hospitable in terms of us. And then eventually the Earth, as organisms have molded it and conditions have changed, it's become a much friendlier place in terms of what we like. But for them, it's not necessarily become as friendly. It's not the conditions that they prefer. But there are still some of these conditions that they prefer, so these guys have persisted and continued on, but they don't tend to live in the same environments as us that often. So I don't know of any Archeans, there might be one or two, but almost none at least, that like cause disease, things like that. So typically we don't worry about Archeans too much. They're more just really cool to study and see how they manage to live in these conditions that pretty much nobody else can. Hope you guys enjoyed it. Take it easy.